Welcome back, gentlemen. Last lecture, we discussed DNA, the structure, as well as how replication happens, so how you get more DNA from a current DNA strand, which is important when it comes to cell division, because when you have mitosis happening, we know, of course, that DNA is copied in the S phase, and replication is the process by which that DNA is copied. Today we are going to move on to the other type of nucleic acid, which is RNA. DNA and RNA, they're both nucleic acids. That's the NA part. The DNA, the D stands for deoxyribose, which is the type of sugar that's in the DNA. So if you remember that nucleotides are made up of a sugar, a phosphate, and a nitrogen base. So in DNA, the D is for deoxyribose. That's the type of sugar that's there. So it's deoxyribonucleic acid. In RNA, it's just ribose sugar, so ribonucleic acid. Now, DNA, as you should know, is that's where your genetic code is kept, okay? It's a storage unit. It just stores your genetic information. But the point of having DNA is that it provides instructions to make proteins. But unfortunately, we can't go directly from DNA to proteins. We have this middleman in between, RNA, because DNA is, you should know, stuck in the nucleus. The DNA is held in the nucleus of your cells. But proteins are made outside the nucleus. They're made at the ribosomes and the cytoplasm from a cell. So this RNA, I want you to think about, is kind of a middleman, like a mailman, carrying the message from DNA to the ribosomes to make the proteins. And then from the RNA, they can make proteins or polypeptides. We're going to get into protein synthesis in our next lecture. So, the flow of genetic information. This is your nucleus of the cell. This is your nuclear membrane, okay? Nucleus is in here. That's this, okay? And you've got your DNA molecule. The DNA molecule cannot leave the nucleus, okay? So from the DNA, you go through a process called transcription to make RNA. And this RNA, so notice the DNA here has, remember we've talked about the double helix, okay? It's a double strand. RNA, the type that's made from the DNA, is now single-stranded. And because it has a slightly different structure than DNA, the RNA is then able to leave the nucleus through the nuclear pores and go through the process of translation in order to make a protein, which is what does all of the work. So, how is the structure of RNA different than DNA? Like I said before, it's single-stranded. So DNA has that double helix shape, the twisted staircase, right? RNA is only half a staircase, okay? I already mentioned before, the pentose sugar, the sugar that is in RNA is now called ribose. Again, DNA, the D stands for deoxyribose, which is the type of sugar that's in DNA. So now we've switched out that sugar for ribose. Both of them are pentose sugars, which means they have 5 sides. It's just a slightly different structure. And then the nitrogen-based uracil replaces thymine. So you still have adenine, cytosine, and guanine in RNA. Those stay exactly the same, but now instead of thymine, you have uracil. There are 3 types of RNA. The first one, messenger RNA, is the one that is made in the process of transcription. So the process of transcription means that you're taking the message from DNA and you're making a messenger RNA. Okay? It's like a mailman. It's taking the message, it's carrying the letter from one place to another. So the mRNA carries the genetic information from the DNA to the cytoplasm. So that is what is allowed. It's made in the nucleus of the cell, and then it can carry that message to the cytoplasm and the ribosomes of the cell. Transfer RNA or tRNA is what reads the message. Okay, so tRNA, the transfer RNA molecules will read the message that's in the mRNA and bring the proper amino acids, which are the building blocks of proteins. They'll bring the correct one in order to put them in the correct order based on the message that's in the mRNA. And then ribosomal RNA is the type of, so it helps make up the ribosomes, which is where protein synthesis actually happens. We're not going to get too far into ribosomal DNA. We're really going to focus more on mRNA and tRNA. So the process of transcription. Like I already said, DNA can't leave the nucleus, but it needs to be able to get the message that it's holding. It needs to be able to get that information somehow out to the ribosomes. So that is what that's what the function of the RNA is to deliver the message from the DNA to the protein or to the ribosome. Now the steps, very, very similar to the steps of replication. Okay, so if you remember, just a little reminder, replication, the DNA unwinds and is unzipped, it's separated, and then you have new DNA nucleotides matching up to the complementary base pairs. RNA is transcription, very, very similar. The DNA separates into two single strands. Now the one side of the DNA serves as the template, and RNA polymerase is an enzyme. You should know that it's an enzyme because it ends in ASE. And RNA polymerase, it's named for what it does. What it's doing is building an RNA polymer, a large RNA molecule, by reading the DNA and bringing the correct RNA nucleotide to pair with it. So when you have an A, we have a modified base pairing rule. So we all know in DNA, A pairs with T, C pairs with G, and vice versa. Now the only difference is that when there is an A in the DNA, we're not putting a T in. We're putting a uracil in. Uracil is replacing that. All of the other base pair rules remain the same. So if you have a C in the DNA, it's going to be a G in the RNA. If you have a G in the DNA, it's going to be a C in the RNA. If you have a T in the DNA, it's still going to be an A in the RNA. The only difference is that if there's an A in the DNA, that is going to now be replaced with a uracil, okay? So that's the only thing that changes, okay? How can you tell the difference how do you know for sure if you're just looking at a string of letters, how do you know if you're looking at DNA or RNA? The main thing is if you have a T, you're looking at DNA. If you have a U, you're looking at RNA. So then RNA polymerase moves down the DNA, it brings in the proper base pair of an RNA molecule until it reaches a termination signal, which is obviously that's the, okay, stop, we're done. And then transcription ends, the newly formed RNA molecule separates from the DNA, and the original DNA strands that were separated now come back together to form a completely formed DNA molecule. So here's up close, like I said, here are those base pairs. This, and this are all exactly the same as in replication. The only difference is that if you have an A in DNA, the one that's going to pair with it is uracil, okay? So take a look at this right here. This is what we call a transcription fork. So not all of the DNA is going to be transcribed at once. It's just going to be a specific segment of the DNA that codes for a particular, it's a particular gene that codes for a specific protein. So it starts at something called the promoter. That's like the segment where the RNA polymerase would be like, okay, yes, this is where we need to start. So it's going to start reading. So the DNA separates. So now you have a DNA strand here and a DNA strand here. And those nitrogen bases are no longer bonded together. They're separated. Okay. One side of the DNA, in this case, that would be this side, acts as a template. So you can see here, there's a T in this DNA, so the RNA polymerase is going to bring in an A. And there's an A in this DNA, so the RNA polymerase is going to bring in a U. And it's going to keep building until it reaches that termination signal. And once it reaches the termination signal, so you see here how the DNA is still open. Once it reaches the termination signal, look what happens. The DNA becomes fully formed again, and the only difference now is that you have this completed RNA molecule that can now leave the nucleus. That's it for transcription. There is a worksheet for you to complete on transcription that will be graded, and then next class, the lecture will be finishing up this whole process with protein synthesis.